My name is Azim Surani. I'm a developmental biologist. I've been working on various aspects of mammalian germline development, which are cells which give rise to sperm and eggs. I was born in Kenya. I was born in a very small town on Lake Victoria. The thing that was of great influence on me was that there were a lot of very exciting fish in the lake. These are called cichlid fish. There are many different varieties. And that was kind of sparked an interest in trying to understand how living things develop. Key always is to find a question. You have to find a question and then also to ask, why is this question important? This I found kind of amazing, that what started as a very esoteric, very fundamental question, like can you make parthenogenetic mice, has a huge impact broadly. So let me explain what parthenogenesis is. It's development of an egg without fertilization, so they don't have any paternal contribution. So the question was, can we do this in mammals? Can we get development of a mammal, in our case a mouse, to develop without fertilization right up to adulthood? So I was working on this with my friend Matt Kaufman, and we could develop these embryos, and occasionally we could even get them to develop up to mid-gestation period, halfway through development, but they wouldn't go further. What we did was to make 3 different types of one-cell eggs. In one, we had 2 paternal derived genomes. In one, we had 2 maternally derived genomes, and the third was a control, it's both. We found that the embryos with 2 maternal genomes had a relatively good embryo but very poor placental tissue, whereas the ones with 2 paternal genomes had very poor embryo but relatively better placental tissue. So we got an answer that, there was something that was happening, this non-genetic information that was coming in. And it was not clear how to describe this. And so when I wrote my paper in 1984, I decided to call it genomic imprinting. In other words, the idea was there was this non-genetic information, if you like, which was imprinted on these parental genomes, which was dictating their functions. We started to think that DNA methylation could be the imprint coming from with eggs and sperm. It's clear that this whole process must be occurring during the formation of the lineage which gives rise to sperm and eggs. One of the key changes that occurs at this time is the erasure of imprints. So by the time these germ cells, primordial germ cells, get into developing gonads, imprints are erased. So the slate is wiped clean. It's almost like wiping out a computer disk. You're going to put on a new program, you wipe out the disk. Now, the embryo can develop as a male or a female. And so they have to put appropriate marks depending on whether the embryo is going to become male or female. And you cannot do that without wiping out the disk or the imprints from these germ cells. And by the time they get into the developing gonads, we call this epigenetic ground state, which means that this is a state where certainly all the DNA methylation is virtually wiped out. The question for me now, which I've been struggling with, is on the evolution of genomic imprinting. I've been wondering whether genomic imprinting had an impact on allowing this diversity of form that you see in mammals. So that's the question that I'm interested in. In this lecture, I'm going to share my journey from Africa to a career in life sciences in the UK. So the work that I've done has been on what I call germline epigenetic imprinting and regulation of mammalian development. So to start with, tell you a little bit about where I came from and how I ended up in the UK. So I was born in a small town called Kisumu near Lake Victoria in Kenya, which is about 30 kilometers south of the equator. So in this picture on the right, you see me with my father around 1950. And behind us is this, what we called our tin house. This was where I was born. It's a one-room tin house. And that's where I started my life. So the thing about growing up in this small town called Kisumu, Lake Victoria, was that it was beside this lake. And one of the things about this lake was that there was stunning sunsets, and also the lake contained many different life forms, including hippos and cichlid fish. Now, the cichlid fish, there are about 500 species, and they evolved very rapidly. And this evolution is driven by what's called adaptive radiation. And I didn't know anything about it. We used to go and catch these fish, and they were all different colors and so on. So it was exciting, but I didn't know what the relevance of the diversity of this species. And what I now know from some of the work that's ongoing in Cambridge, that this is a good model to understand evolution of vertebrates. So I went to a school in Kisumu High School. This is the science block here. And it was really here that I was fortunate to have a very good biology teacher who got me interested in diversity of life and the different life forms. So that kind of sparked my interest in living organisms. So around 1963, this was a period of some uncertainty because Kenya was approaching independence. It was formerly a colony of UK, British colony. So in 1963, it was it became independent from Britain. So because of this uncertainty, my parents decided to send me to UK, and they asked me to go and get education because it was necessary for my survival. So I went to UK and I went to a couple of universities. The situation in the UK was slightly difficult because there were many immigrants coming from various colonies. So I decided to leave UK and I went to France for a couple of years. And when I was there, I had a chance meeting, and that kind of changed my life completely. And I ended up in Cambridge in 1972 for a PhD. So my fortunate break was meeting Bob Edwards. He's the pioneer of test tube babies, or IVF. And in fact, he got a Nobel Prize for his work in 2010. So when I got to Cambridge, he asked me to work on implantation of embryos, implantation of blastocysts in womb. This is a very early stage before the embryo starts to grow. And he wanted me to work on the mechanism of blastocyst implantation in rats because this event was one of the major hurdles for development of in vitro fertilization and success of test tube babies. While I was working on this project, I became more interested and fascinated by the development of the embryo themselves. So that's what I got interested in. And in any case, Louise Brown, the first test tube baby, was born in 1978. So I thought that in principle, this problem had been solved. So I wanted to do something different. So I'd met a colleague called Matt Kaufman, and he was working on this project, which was to take mammalian eggs, unfertilized eggs, and what he was doing was to artificially activate them So they didn't have any paternal genome, but he could make these kind of little one-cell embryos or zygotes with two maternal genomes. And the work that we were doing, we found that these embryos could develop sometimes quite far, up to about mid-gestation, halfway through pregnancy, up to 25 somite stage. And so this looked very interesting. But the question was, can they develop to adulthood? Can they form into living, free-living organism? Now, this question was interesting because there are some vertebrates, like this lizard here, which can develop what's called parthenogenetically. So these are unfertilized and they can reach adulthood without fertilization. There was quite a strong public interest in this area, and there was a lot of debate about whether sexual reproduction was necessary in mammals. And there were books, like this book was written around that time called The Redundant Male. So there was a lot of discussion around this area, which probably also was responsible for stimulating my interest in this question. So really my question was, are paternal chromosomes necessary for mouse development? In 1979, I got the opportunity to start my own laboratory at this institute called Animal Research Station, which is just outside Cambridge. So I joined that in 1979 with my assistant, very experienced embryologist. And she was really quite vital to the success of the work that we did. So I started my work at the Animal Research Station, and after many experiments over a few years, we found that we couldn't get asexual development of mice to term, to adulthood. So to cut a long story short, and to get to the next part, what we did was to make three types of embryos. We made embryos which contain a male and a female genome, or chromosomes. We had made another construct here, which only had two female or two maternal chromosomes, from two different females. And the third one where we had chromosomes from two males. So these are three entities which are genetically identical. And what we found was that only those entities which had a female and a male chromosomes were able to develop into viable adult organisms. The two embryos with two maternal chromosomes, or these entities with two paternal chromosomes, could not develop. They died during development and didn't reach their full development. Now, this is an interesting experiment because theoretically, these are genetically identical, so they should all develop. They should all have the potential to develop according to the Mendelian laws of genetics. But only one of them could develop, and it required the presence of both maternal and paternal chromosomes for normal development. Around this time, the group of Davos Salter also found similar results. So the reason why you require both these parental genomes became evident in the next experiment, and was really led to the discovery of what we now call genomic imprinting. What we found was these genetically similar parental genomes exhibit functional differences due to epigenetic asymmetry. So although they are genetically similar, they are epigenetically different. So this genomic imprinting phenomenon is epigenetic modifications to DNA that are inherited through sperm and eggs without changing the DNA sequence itself. And how these functional differences manifest can be seen from in this experiment, where you see that if these entities contain only the paternally derived chromosomes, they can form a reasonably good placenta, but the embryo is quite poor, whereas those entities with two maternally derived chromosomes can develop relatively good embryo, but very poor placenta. So this shows that there are these functional differences between the two parental genomes. So we wanted to kind of understand the mechanistic basis of how these kind of differences come about.
Yeah.
So that's kind of shown here. So as I told you that the parental genomes, the male and the female, are epigenetically asymmetrical, although they're genetically similar. And I want to take you through this experiment, which we're just looking at one chromosome. This is chromosome 7, to illustrate this phenomenon of epigenetic asymmetry between the male and the female chromosomes. So here we're looking at the chromosome 7. This one here on the left is the copy that has come from the father, and the one on the right here in pink is a copy that's come from the mother. And what I want to point out is that here you see on the left, there's genes that are shown in blue. These are genes that are only expressed from the chromosomes that has come from the father, whereas these genes here on the right, shown in red, are expressed from chromosomes that's come from the mother. So you can see that these chromosomes, parental chromosomes, are expressing, at least some of the genes are expressed differently between the two chromosomes. So to get to the next level of understanding the molecular basis of these differences, I want to concentrate now on this region here at the end of this chromosome 7, which contains these two genes, IGF2 and H19. So what I want to show you here is that on chromosome 7, there are two genes. One gene is called this H19, and the other gene is called IGF2. So this is the two copies here of IGF2 and H19 here. Between these two genes, there's this region called imprinting control region here, which are shown here at the end. And what I want to point out is this marks here, these lollipops with black dots that I've shown, is the signature or the imprint signature. And this depicts the methylation of CpG dinucleotide. So it marks the imprint, which in this case comes on the maternal chromosomes. And as a result of this imprint, what happens is that these regulatory elements on the maternal chromosomes will induce expression of H19 gene. But these regulatory elements on the paternal genome will induce expression of this gene called IGF2. So you can see that on this chromosome 7, you have this result where on the maternal chromosomes, it is the H19 gene that is expressed, and on the paternal chromosome, it's the IGF2 gene that's expressed. So this is kind of the principle that shows that this imprint can influence expression of parental alleles. So you get this kind of monoallelic expression of one of the parental alleles. In this case, H19 on the maternal and IGF2 on the paternal chromosome. So this is just one illustration, and there are variations on this kind of theme. But on other chromosomes, there are also similar mechanisms that are regulating expression of these imprinted genes. So if you now go forward, what I want to point out is that there are around 200 imprinted genes, which is approximately 1% of the genome. There are about 30,000 genes in mammals, and 200 of them show these imprinting expressions. So only one parental allele is expressed. And these imprinted genes have diverse functions. They regulate development, growth, physiology, brain development, behavior, metabolism. So they regulate diverse functions. They don't regulate just one particular function in mammals. IGF2R was the first imprinted gene to be identified by Dennis Barlow in 1991. And then many other imprinted genes were also identified. And we did some work where we also identified other imprinted genes as well. So just to emphasize, this epigenetic inheritance from the germline does not change the genetic blueprint, but it influences which parental allele is expressed. I'm going to give you two examples of expression of the functions of two imprinted genes. This is one gene. This is retrotransposon-derived, what we call PEG10 gene, which is crucial for development of the placenta. The interesting thing about this gene is that it originated 160 million years ago and is only present in eutherians, that is us, and the marsupial mammals. And this gene was found to be very important for the development of the placenta. This gene is expressed, only the paternal copy is active, the maternal is silent. And what was found was that a mutation in this PEG10 is lethal because it prevents the development of the placenta, so the embryo does not progress. It dies in utero after some time. So this is one example of the function of imprinted gene. The next example I want to show you is two other genes called PEG1 and PEG3. These are linked to brain development and mammalian behavior. So here is PEG1 gene shown expression in the cortex and the hypothalamus. And the PEG3 gene very similarly shows expression in the developing mouse brain. And what was found was that a mutation in PEG1 gene causes abnormal maternal behavior. So what this is showing is that this female does actually produce young, and they're born, but the mother shows defective maternal behavior, so she doesn't build a nest and doesn't look after the pups after they're born. So the pups are fine if they're rescued and put with foster parents, then they will survive. But this mother, which is a mutation in PEG1, doesn't take care of them. So this shows that in this particular case, these imprinted genes have an impact on maternal behavior. So these imprints that are put on during development of the eggs and sperm are heritable. So these imprints are passed on after fertilization right into adulthood. And there's a mechanism that recognizes these imprints. And these are two factors here, shown here, ZFP57 and ZFP445, which recognize this imprint. And they transmit these imprinted genes through to adulthood, where they have diverse functions in developing embryos and in adults. And as I said before, they regulate growth, differentiation, cell fate, metabolism, neuronal functions, and so on. If imprinting goes wrong, it gives rise to approximately 14 human diseases. So I've told you about the initial work on imprinting, where we found this phenomenon of genomic imprinting based on the embryological data. And we have come to the point where we're starting to understand the molecular basis of how this system actually works in mammals. So in 1992, I moved back to the university where I got elected as the Marshall Walton Professor and a Fellow of King's College, Cambridge. And I started teaching natural science and medical students. And I started to expand my research on imprinting, epigenetic, germline, and also on human development. So that was the next phase of the research that I was getting into after I moved to the university in 1992. So I told you about this epigenetic asymmetry which exists between the maternal and paternal genomes at fertilization, which is a basis for these functional differences. Now, this epigenetic asymmetry is actually programmed during the development of the germ cells, which is the cells that give rise to sperm and eggs. And I want to tell you a bit about that now. So in humans, the primordial germ cells originate around the second week of pregnancy. So they're quite early, because these are the cells which are going to give rise to the next generation, and in fact, subsequent generations as well. So these are the most important cells. They form very early during human development. And what happens is that as these cells start to proliferate and develop, they start migrating from this site here where they originate. And they migrate towards the developing gonads in the fetus. And by the seventh week of human pregnancy, these primordial germ cells have entered the developing gonads, and they start to undergo differentiation into sperm or eggs, depending on the sex of the embryo. While these germ cells are migrating, the first thing that happens is that the imprints that I talked about, first of all, they're all erased. So this is the start of the resetting process. They're erased, and then later on, when sperm and eggs start to form, new imprints are introduced, depending on whether this is sperm or eggs. So they have different imprints are put on depending on we are looking at development of the sperm or eggs. So I want to tell you a little bit about how this process occurs. So let me go back to this point here. I told you that these primordial germ cells, the precursor sperm and eggs, develop around the second week of development, second week of gestation. So what we found was these are the key genes here marked in red, which are important for the initiation of this formation of this group of cells, the primordial germ cells. So this is what we call the genetic network, which governs the formation of these very early primordial germ cells. And this genetic network has three important functions. So these genes induce formation of primordial germ cells, and at the same time, they repress the cells from adopting other fates, so body cells like somatic cells or any other cell type. So they're confined to becoming only primordial germ cells by this network of genes. The other key thing that happens with these genes is that they initiate this epigenetic program, which is going to erase and reintroduce new imprints. So this network is really quite crucial, and we've done quite a lot of work on this. So here what I'm showing you is another way of depicting this process of resetting. So here is the point where these primordial cells are formed, and as they start migrating towards the gonads, we get erasure of imprints. In fact, global DNA demethylation occurs, which declines to very low levels. And just to point out one aspect of the mechanism, the network that I showed you represses this gene called UHRF1. UHRF1 is repressed in this migrating primordial germ cells. And as a consequence of this repression of UHRF1, this prevents methylation of DNA as it replicates. So essentially, this repression of UHRF1 ensures that we get erasure of DNA methylation. So this is one aspect of the resetting process at the molecular level. So I'm depicting this another way. This is the reset, what we call the reset switch, which initiates primordial germ cell specification and this epigenetic program as the primordial germ cells enter into the gonads. And as they start to develop into sperm and eggs, there's this reestablishment of imprints. And then at fertilization, you recreate this epigenetic asymmetry. So this is process is reset for each generation. It doesn't persist forever. It's just for each generation. So the erasure of epigenetic memory and imprints is an important part of this first part before this new imprints can be introduced. So what I showed you initially with the mouse work, we also see in humans, and this is, I'm now showing you what happens in humans where these kind of entities occur naturally in some cases. It's at a very low frequency. So here, what I'm showing you is that when you have genomes, chromosomes from female mothers and fathers, you get this normal development of the fetus and the placenta, you can see here. But when you have chromosomes only from two sperm, this egg is fertilized by two sperm and you have two paternal chromosomes with no maternal chromosome, you get what's called hydatidiform mole. And then what you see here is they only get development of the trophoblast or the placental tissue and completely lacks fetal tissue. So you can see that if you don't have a maternal genome, there's no fetal tissue at all. With two maternal chromosomes, you do get what is called ovarian teratoma, which these eggs don't actually develop in utero because they lack the placenta. They sometimes stay in the ovary and they form these ovarian teratomas, and these contain only fetal tissue. They don't have any placental tissue. So again, you can see here that these chromosomes from mother and father have these differential functions because of imprinting, and they also have this epigenetic asymmetry, which I described for the mouse experiments. So, as in mice, this genomic imprinting is essential for human development and health, and you can see that these imprinted genes have diverse functions in humans as well, regulating growth, control of placental and fetal development, cellular proliferation and differentiation, energy homeostasis, brain and behavior, endocrine function. So there are a number of functions these imprinted genes have. Now, if imprinting goes awry or if there's a mutation which affects imprinting, I'm showing you here an example of disruption of imprinting on chromosome 15. It results in a syndrome called Prader-Willi syndrome. And the phenotype of this Prader-Willi syndrome is that there is delayed puberty, there is obesity due to increased ghrelin, and there's type 2 diabetes, there are behavioral issues. So Prader-Willi syndrome occurs partly because of this defect in imprinting on chromosome 15, and it affects this gene SNRPN amongst others. So this is just one example, as I mentioned before, there are 14 human diseases which occur when you have 40 imprinting. Now, there are experiments that are ongoing in trying to reverse this 40 imprint. And one of the things that is being tried is to see, as I told you, there are two copies of genes, one from mother and one from father. So one copy in this case is silent due to this methylation. And there are some experiments that are being tried which is to try and remove this methylation or to demethylate this imprint to allow restoration of expression of this SNRPN gene. So this is just experiments in culture at the moment to see if it might be possible to activate this repressed copy of SNRPN and other genes to rescue this Prader-Willi syndrome. So there's ongoing work to try and attempt to address this problem of imprinting defects. I think the other question that is really very interesting is imprinting and mammalian evolution. Now, imprinting originated around 160 million years ago in the Therian lineage, before the split into marsupials and eutherians, the rest of the mammals. Around 60 years ago, there was mass extinction where most of the dinosaurs were wiped out. And what happened then was that the eutherian mammals underwent adaptive radiation, very similar to the adaptive radiation I described in the cichlid fish in Lake Victoria. So you start to see diversity of mammals, and particularly eutherians showed extreme diversity of mammals following the loss of dinosaurs here. And they started to occupy different niches that will become available for mammals to diversify and occupy the land. Similar process occurred in marsupials, but to a lesser extent. Now, there's a third group of mammals. These are monotremes. These are egg-laying mammals. They don't have imprinting. And you see that they don't have the same degree of adaptive radiation, which you see here. So it's possible that imprinting had a part to play in this adaptive radiation mammals and the existence of the diversity of mammals that we see now. Of course, what this imprinting did was that it was responsible for changes in the strategy of development. I'm just going to show you one example which affected very early development. So here are the monotremes, which are the egg-laying mammals. So you can see the development of these embryos. So I'm showing you development of one egg, the zygote, the one-cell embryo, through to blastocyst stage before implantation. So these are very yolky eggs we see in monotremes. When you come to these placental mammals, which are viviparous and give rise to live young, you can see that there are changes here. So you see in eutherian mammals, in us, we don't have these yolky eggs. But what happens is that these embryos start to develop, they start to proliferate, and they form these enclosed cells, and they form blastocysts. And here you see a very distinct group of cells. These are inner cell mass here, and the outer trophectoderm cells. So the inner cells will give rise to the fetus, and the outer cells to the placenta. And this is where the imprinting has an impact. So the maternal genome will have an important role in the development of the embryo from this ICM, and the paternal genome development of the placenta from these outer cells. Marsupials shows to some extent similar trend as well, but they have some differences. They don't have this multi-layered blastocyst. The blastocyst is a single layer of cells. So there are some differences between the marsupial and eutherians, although they both have imprinting. So finally, this is the final slide to summarize what I've told you. I want to tell you about the impact of genomic imprinting on life sciences and medicine. So I told you that this imprinting resulted in epigenetic asymmetry and functional differences between the two parental genomes. So one of the key outcomes from this study, is that we see that there is inheritance without changes, without genetic changes, which is the epigenetic inheritance. So this epigenetic inheritance is reversible, and it is reset in the germline for each generation. It doesn't continue from one generation to next. It needs to be reset. This provided a paradigm shift in epigenetics, was the foundation of epigenetics research. So this is a non-Mendelian inheritance, and which is alongside genetic inheritance in mammals. So this is an important component of inheritance and non-genetic inheritance. And it's important, as I showed you, for development, and faulty imprinting can give rise to human diseases. As I showed you, these functional differences are responsible for development of the placenta in the fetus. And they also regulate growth and differentiation, development of the brain and behavior, stem cells and regeneration, and faulty imprinting causes human diseases. I think it may also have played a part in evolution, give rise to what we call placental viviparity, which is development of a fetus in utero. And we know that in Eucerian mammals, the gestation period can be very long. In some mammals, it can be as long as 2 years. So this long gestation period might have been important for eventually for giving rise to the diversity of mammals and to the adaptive radiation, which I tried to describe. So the diversity of mammals we see now may in part be due to genomic imprinting. Thank you.